members and delegates. Greetings from Neonatology Forum, Kerala. I am Dr. Ajobalan, President, Neonatology Forum, Thrissur. Let us start the last webinar of Learn from the Legends of 2022 series. This international bi-monthly webinar series is an academic initiative of IAP NeoChap, which is organized by NNF Kerala and IAP Thrissur. We have nearly 9,300 registrations from 75 countries for this scientific feast. A warm welcome to all the delegates in Zoom platform as well as YouTube channel on behalf of IAP NeoChap and NNF Kerala. Today, we have an eminent faculty, Professor Michael Georgieff from University of Minnesota, USA, to address an important topic, what's new in nutrition and brain development in neonates. We welcome you, sir, for the webinar.
Thank you so much.
Two experts in neonatology of our country are moderators for today's session. Dr. Colonel G. Shridhar, Professor of Pediatrics from AFMC Pune, and Dr. Colonel Subhash Chandra from Army Hospitals, RNR, Delhi. Hearty welcome to both of you for the webinar.
Thank you, sir.
Now I request Dr. Manoj VC to introduce the chief guest.
Hello, everyone. I am Dr. VC Manoj, and on behalf of Team Learn from the Legends, I thank you all for joining us today. Today, we have a very popular researcher as legend of this month, Professor Michael K. Georgieff. Let me welcome him, the moderators, and all of you to this last session of 2022 in this third year of Learn from the Legends International Neonatology Webinar Series. Professor Georgieff is the Martin Lenz Harrison Landgrant Professor of Pediatrics at University of Minnesota, USA. He is also the Executive Vice Chair of Pediatrics and Co-Director of the Masonic Institute for the Developing Brain at the University of Minnesota. Michael is an internationally recognized expert on the effects of nutrition on the developing brain and specifically the effects of iron status on learning and memory processing. In fact, his career in this area has spanned over 35 years and includes investigations of brain function in humans and preclinical models, which I'm sure we are going to hear about today. Professor Michael has also conducted multiple studies on the effect of fetal and neonatal iron deficiency and neonatal anemia on the developing brain regions and behavior. Needless to say, he has published over 280 peer-reviewed papers and was a consultant to the National Institutes of Health about future directions in cognitive research. In fact, Professor Georgieff is also a member of the prestigious BOND, Biomarkers of Nutrition for Development, IRON, and BONDKIDS Working Group for NIH and NIH Global Health consultation work groups. Over and above all, I have realized that he is a very humble human being. Many of you who had attended Hippocrates last month at Bangalore and had interacted with him, I'm sure will agree with me on this point. So friends, without much ado, let's hear from this legend's mouth. Over to you, Professor Michael.
Thank you, Dr. Manoj. I certainly appreciate that. It was good to see you at Hippocrates. And for whoever is out in the audience who has seen this, who was there as well. Let me share my screen. Let's do that first. And we'll put that in presentation mode. So nutrition and brain development, an important topic that, unlike many other aspects of brain development that we have in the newborn intensive care unit, we actually have control over this one, something we can actually do something about. And so that's why I think nutrition is such an important piece of what we do for the babies, both term and as well as preterm babies. It's a huge topic, and I don't pretend that we're going to be able to cover the entire waterfront in a single lecture, but I know we have plenty of time for question and answer afterwards. So I think we can hone in then maybe on more specific areas that you would like to talk about. The objectives of the talk are really just to define the principles of nutrient and brain interactions. In other words, what are the rules of engagement? I think so often in our research, in neonatology in general, we say there is a cause and there must be an effect, but lots of times there's lots of hand waving that goes on. There's some very specific principles that apply to any sort of environmental interaction with the developing brain, and nutrition is a good example of an environmental factor that affects brain development. Then we want to, once we've kind of established those ground rules, we want to get into identifying which specific nutrients we need to be concentrating on. In other words, all nutrients are important for the brain, but there are some that are much more prominent and have much more specific effects, more profound effects on early neurodevelopment. And finally, we need to understand that how we treat babies nutritionally early on not only has an effect on how they respond physiologically while they are in our hands, but also long-term. Nutrients have a way of imprinting genes and imprinting structure in the brain that has lifelong ramifications. And that's really the cost to society when we don't do that right. So we'll be spending some time on those mechanisms as well. So I've already kind of alluded to this issue that early nutrition is important now, meaning for the baby at hand, but is also important in terms of determining or driving long-term relevant health outcomes. This comes from an area of work called the Barker hypothesis or the developmental origins hypothesis, but is important because it links early nutrition to what are called relevant health outcomes, which I've listed on the left here. Barker's original work, or the developmental origins original work, really had to do with intrauterine growth and then cardiovascular outcomes 60 to 80 years later. But there are other relevant health outcomes that are imprinted or affected by early nutrition, and they include metabolic health, immune health, bone health, risk of cancer, and now we know brain health as well. There's an interesting, I'll refer you to this article that is on the right here, and you'll see some very familiar names there, including the very eminent Dr. Bill Hay, Dr. Joseph New, who was the one who led the Hippocrates convention recently in Bangalore. All of us were part of a working group that was evaluating how early nutrition, particularly in preemies, so that's what PRE stands for, the Premature to Birth Project, PRE-B project, and how it affects relevant health outcomes. So that's what we are concentrating on. Now, there's no question that early nutrition in some way or shape or form affects long-term neurodevelopmental outcome and mental health. One of the longer standing studies comes from Finland, and this just shows you that growth from birth in preterm babies, growth from their birth to term, has an effect on their IQ, their verbal IQ, their performance IQ in the teenage years. Whether we're talking about growth as defined by weight, as defined by length, or as defined by head circumference. So each one of these has a positive effect on neurodevelopmental outcome long after the babies have gone home. In other words, at teenage years. And in fact, the scaffolding that takes place affects not just general neurocognitive abilities, in other words, IQ, but also executive function, attention, and factors that are often not measured in the IQ, but still are very important for success in school and ultimately success as a worker. So I think it's been clear since the 1980s, a very prominent professor, Richard Ehrenkrantz, working with the NICHD network, probably showed it the best, that poor neonatal growth, in other words, lagging in weight, in length, or in head circumference, any of those is associated with poor neurodevelopmental outcome. And as I said, that literature goes back a long time. And more recently, studies by Professor Cook and Embleton and Rigaud have shown that interventions, macronutrient interventions, so calories and protein, will improve growth in premature infants. So we know that poor growth is associated with poor developmental outcome, and we know that intervention can make a difference in growth. And the second bullet point here is, that not only is that true for macronutrient, but it's true for micronutrients as well. In other words, deficiencies of certain micronutrients, think about iron, zinc, iodine, will negatively affect brain development. And we also know that micronutrient supplementation improves the micronutrient status. So that's clear. What is much less clear and what has been much more difficult to prove is whether interventions or that improvement in preterm infant nutrition status affects long-term relevant health outcomes, and specifically neurodevelopment. In other words, there's not a lot of studies out there where an intervention randomized kids to more nutrition or less nutrition, whether that has a significant impact on neurodevelopment. And currently, I think that's where the field is at, is gathering that information, and particularly in preterm infants. So currently, the way we think about it in the unit when we take care of babies is that growth and nutrient status, in other words, what we measure in terms of length, weight, head circumference, or for that matter, serum levels of nutrients, act as surrogate markers for neurodevelopment. In other words, if we can improve the nutritional status, the neurodevelopment will follow. And I think that's a perfectly reasonable way to think about it. I just wanted to point out it's been very difficult to actually show that the interventions make a lot of difference. So let's think a little bit about why this might be the case and go through just the principles of nutrient-brain interactions. So nutrients regulate, there's no question that they regulate brain development and function. I'm going to show you a slide a couple ahead here on biochemically, or actually cellularly, exactly how that works. But I think we need to realize that, first of all, nutrients are not simply fuel. I mean, it's kind of easy to think about it that way, but nutrients are also involved in generating growth factors. And if you think about a car, you really need both, right? You need fuel for the combustion of the engine, but you also need an engine. You need a transmission there to literally transmit the fuel into positive energy, into positive motion for the car. In this case, growth for the baby. So you will get reduced growth and function of the brain if there's insufficient nutrients, so not enough fuel, insufficient nutrient amounts, we're not giving enough, or accretion. We have things that are inhibiting the uptake of nutrients or the utilization of nutrients. And I'll just point out that one of the most important ones of that is infection. Infection diverts nutrients from being taken up and used for tissue accretion, and instead sends it through the gluconeogenesis pathway for basically a fight or flight phenomenon. So either insufficient nutrient, insufficient fuel, or suppressed growth factors. So in other words, you could have plenty of fuel, you could be giving a baby plenty of food, and yet if the growth factors are suppressed, as happens with, say, zinc deficiency or with stress or infection, you will not get adequate growth. Similarly, you could have plenty of growth factors, but not enough fuel, not deliver enough, and again, you will compromise growth. That rapidly growing brain that we are talking about here is much more vulnerable to nutritional deficiency than, say, a child of age 5 or 10 or older. And that's because, and we'll go through this, it's because the brain is so rapidly developing, things are changing so rapidly in terms of dendritogenesis and synaptogenesis and myelination that you miss critical periods of development if you're not giving adequate nutrition for a certain amount of time. Now, the counter side of that coin, which is truly amazing about babies, and those of you who do NICU follow-up know this, that the neonatal brain is remarkably resistant or remarkably able to repair itself after injury. Certainly babies with large hemorrhages have their issues long-term, but I will tell you that in my NICU follow-up clinic, I have babies who have a stroke, for example, in the newborn period, and 4 months later, I can't tell which side the stroke happened on. And that's the ability of the baby's brain to rewire itself. All of that said, this business of vulnerability versus this plasticity, I think everybody in the field agrees that vulnerability outweighs plasticity. And that's just a simple way of saying, stay on your growth curve, continue the developmental trajectory rather than interrupting it and then expecting catch-up growth, for example, to repair the damage. I mentioned in the previous slide that the cellular biology of how nutrients affect brain development is pretty well developed, and there are several signaling pathways in all cells. I'm happening to be showing this as a brain cell that take nutrients, and they are found out here, right? You can probably see my cursor, and then you will see this double line. That's the cell wall, and there is a signaling pathway here called the mTOR system, which is able to sense critical nutrients and how they are and their availability. So, for example, protein intake, and more specifically, branched chain amino acids are conveyed through signaling factors here that are shown in green. Growth factors that we talk about, like brain-derived neurotrophic factor or IGF-1, signal through this PI3K signaling pathway. Iron and zinc can affect BDNF levels and IGF levels, but iron also works through this HIF-1 pathway and this AMPK pathway, as do glucose and calories. Bottom line is that the brain cells have the ability to sense nutrient availability through these receptors and through these signaling pathways. And look what comes out the bottom, and I've circled that in red. You get cell size and survival. So first of all, at the top of the algorithm, do I have enough nutrition as a brain cell to survive? And then secondly, how big am I? How complex am I? How much actin organization do I have? What rates of translation and transcription do I have? So you can see that the brain, as it's building itself, uses these nutrients to make itself more complex. And there is plenty of data that shows that the more complex the dendritic arbor is and so on in various areas of the brain, the more capacity it has, and therefore the better or more mature the behavior is. I'd like to point out that while we think of the brain as mostly made up of neurons, and neurons are certainly the workhorses, and you mostly think about anatomy because that's what we can see, nutrients actually affect many different processes in the brain that are important for the function of the brain. And ultimately, that's what we want, right? We want function. Anatomy is interesting, but it's function that we're after. So examples here. Yes, nutrients can affect neuroanatomy, but that is made up not just of the neurons themselves, but the supporting cells also have high energy needs. That includes oligodendrocytes, astrocytes, microglia, and you can see what their jobs are. For the neurons themselves, early on nutrition, and this is mostly fetal, you are affecting the number of cells that you have. But for the most part, when we take care of the babies, most regions of the brain have post-mitotic neurons. And at that point, we are talking about growth, so size, and most importantly, complexity. And there are certain nutrients that support these processes more than others do, and we'll get into that in a second. However, anatomy is kind of inert, kind of a lump, just sits there. You actually need to put that anatomy into motion. And of course, we know that happens through neurochemistry, or what we would call neurotransmitters. And here, nutrients are important for the concentrations of certain neurotransmitters. An example there is iron. Iron is intimately involved in the synthesis of dopamine and serotonin. It also affects glutamate and GABA. Choline, for example, affects levels of acetylcholine. So certain nutrients are involved in determining the concentrations of neurotransmitters, as well as the regulation of their receptors, their expression on the postsynaptic membrane, and the reuptake by the presynaptic membrane, the recycling. How quickly can the system recover to fire again? Again, neurochemistry is interesting, but it has to generate an electrical potential. And that's where a lot of the energy demand of the brain comes from. Neuronal metabolism that drives the electrical activity of the brain is highly energy consuming. Right? So realize that nutrients are not affecting just how the brain is being built anatomically, but also how it functions. Now, for every cell in the body, and that includes every one of those types of brain cells, all nutrients are important, all 23 or 24 of them. But there are certain nutrients that have particularly large effects on the developing brain and their function. And I've listed them in three kind of categories here. And the way I've listed them also with the superscript tells us about the processes I just talked about. In other words, which one of these nutrients it has its large effect because it affects neuroanatomy, either the neurons or the glia, which ones affect the neurochemistry, the neurotransmitters, which ones affect the ability of the brain to actually perform its function, that is the electrical potential. And the fourth category we haven't discussed yet, but will toward the end, about how it affects gene expression or gene regulation, particularly of what we call synaptic plasticity genes, genes that are involved in regulating how the brain functions. And I've divided these nutrients into conditions. In other words, where there are multiple nutrients involved, like stunting and underweight and obesity, and then macronutrients and micronutrients. And the ones that I've highlighted in red are the ones that we're going to be going into in a bit more depth, mostly because they're either the most common deficiencies, or else they are the ones that have the biggest impact and are ones that we can control. So I've already kind of alluded to this, that, when you have an altered nutrient status, and this applies not just to preterm or to babies, but also to older kids, the altered nutrient status can affect the brain's structure and function, as we talked about. And if it only affects it during the time of the deficiency, let's say you have a child who's acutely zinc deficient or iron deficient or protein deficient, not surprisingly, that brain will not function very well. So we could term that acute dysfunction. Examples of that, it could be as simple as just morning energy intake and your daily school performance, or acute or subacute deficiencies as occur in our NICUs, but that lead to reversible changes in brain physiology. And those effects can be global. They can affect the way the whole brain functions, or very regional, either regionally like an area of the brain, or regionally as in a process like myelination. Very few nutrients actually have unique signature effects on the brain. And what I mean by that is, it would really help us all out if we could look at somebody and say, your behavior tells me that you are zinc deficient, or your behavior tells me that you are iron deficient. Unfortunately, there's very few examples of that. So on the one hand, we can have deficiencies that cause acute dysfunction of the brain, and that could be problematic at the time. But the ones that we're most concerned about, and the ones that when Dr. Manoj read some of the councils that I have been on, the concern is at the population level, the long-term effects that take away potential for school performance later on, and ultimately for job performance and even socioeconomic status. So these are effects that last beyond the time of the deficiency and cause altered development and adult function. So critical periods of brain development exist, and the changes that take place with the early nutritional deficits alter the structure of the brain during these critical periods. And not surprisingly, the cost to society is really from these long-term effects. I love this quote that was from the Lancet a few years ago, that eradicating the iron, zinc, and iodine deficiency in the world would increase the world's IQ by 10 points. And so I might ask for a show of hands of who would like 10 more IQ points today. I certainly could use it personally. Okay, so then let's come back to our list again and talk about these long-term effects. You can see here that same list, but now the superscripts have to do with, these are all nutrients that have been demonstrated to have long-term effects. That is, if you have a deficiency or a change in status early in life and don't correct it immediately, you will affect long-term neurodevelopment and mental health risk. So the ones that I've superscripted 1 here exhibit a critical or sensitive period for neurodevelopment. That means, as I'll show you later, that if you don't have nutrient sufficiency during that critical period or sensitive period of development, you have long-term structural deficits. The second, those that are superscripted with a 2 are those in which early deficiency results in long-term behavioral dysfunction. So that you would think would map pretty well onto those that show that sensitive period. And the ones with the superscript 3 are really the exciting group to me, because they're the ones that show that some of this is not just about poor structure, but about gene regulation across the lifespan. And just a kind of a quick aside about that, not just in the brain health space, but in that cardiovascular space that I talked about in the very beginning, it's evident that the fetus, and of course the baby that we take care of in the NICU, is trying to anticipate what the rest of life will be like. In other words, many enzyme systems, from a cardiovascular standpoint, that would include glucose handling, hepatic enzymes, and so on. In the brain, it means brain-derived neurotrophic factor and certain synaptic plasticity genes. Their regulation is set early in life and remains pretty immutable across the rest of the lifespan, at least in the animal models. And that is regulated by changes to DNA, epigenetic changes to DNA, either histone changes or DNA methylation. We'll get back to that toward the end of the lecture. So let's drill down now on these major nutrients, the ones that have the biggest effect in the neonatal brain. And as bad as a rap as carbohydrates have gotten in the public press, carbohydrates are pretty important. The neonatal brain basically runs on glucose. Now, as you all are listening to the lecture, your brain is using about 20% of your total body oxygen consumption, and your total body caloric need, as it relates to your oxygen consumption, is about 40 calories per kilogram of body weight per day. We all know that babies' nutrient requirements or energy requirements are around 120 calories per kilo per day, and that neonatal brain is sucking up 60% of that. 60% goes to the brain. So you can imagine that when that is shortchanged, that you will have significant energy disruption of the brain, and that has ramifications then on the ability of that brain to do what it's supposed to do, which at that time in life is growth and differentiation. So glucose is the main metabolic fuel for the brain. It can use alternate sources like lactate and ketones, but glucose is the preferred one. Protein, obviously very important because that's the structure of the brain. It's important for DNA and RNA synthesis and maintenance. I will remind that neurotransmitters are proteins and therefore subject to protein status. Growth factors are proteins. And then, of course, the structural proteins themselves, that is the axons, the dendrites, and the synapses, Our current recommendation is that protein be started at 3 grams per kilo per day on the first day in preterm babies and advance to a goal of probably somewhere between 3.5, maybe as high as 4.5, probably more on the order of 3.5 to 4 grams per kilo per day. And then finally, fats are important because they are part of not just myelin composition. I think that's what most people think about with the fatty acids, but also for membrane composition, meaning synaptic membranes that are going to fuse, the neurotransmitters are going to fuse with the outer cell membrane and release them. Dr. Manoj mentioned that I know a few things about iron. I could probably spend the entire lecture on iron, and we can certainly talk about it in depth in the question and answer period. Iron, as you all I'm sure know, is absolutely critical for the developing brain. It's contained in enzymes and hemoproteins that are important in actually various aspects of brain development, including myelination. So some of the desaturases that are involved in the synthesis of myelin or as the composition of myelin are iron dependent. The area of the brain the regions that I study have very high energy needs, and the areas like the hippocampus, the dendritic structural complexity is highly iron dependent. So you have an iron deficient hippocampus, you have structural problems. The oldest literature is in the monoamines. As I had briefly mentioned, iron is important for the enzymes tyrosine hydroxylase and tryptophan hydroxylase that synthesize dopamine and serotonin and norepinephrine. So just think about, the entire reward and feel-good system of the brain is iron dependent. And finally, we've learned over the last 10 years or so, actually not quite even that long, maybe 5 to 7 years, that iron-containing enzymes exist, for example, histone demethylases that contain iron that determine the histone status and therefore the gene expression of multiple individual genes, as well as large gene networks that regulate brain function. So iron pretty important in terms of brain function. Is there evidence that there are neurodevelopmental sequelae of perinatal iron deficiency? Yes, starting with work by Tamura et al. Back in 2002 in term infants, that babies with the lowest quartile of iron, of ferritin that is, less than 76 micrograms per liter, they have poor school-age neurodevelopment. Now, Tamura did not know why that was the case. This was just an observational study. But subsequently, Asha Siddhappa, who is from Bangalore and who is a member of our faculty now, showed in 2004 that term babies who are born with a cord ferritin less than 40 have impaired hippocampal function, impaired recognition memory that persists up to 5 years post-discharge. And then Ted Wax has shown that, yes, there is a dopamine effect. Iron-deficient infants who are born to iron-deficient mothers have altered temperament, poorer bonding, poor maternal-child interactions. And then in our preterms, there are a couple studies now. I've listed the Steinmacher one, but there have been a couple more recent studies on early iron supplementation and better mental processing at older age. Sanjeev Amin showed nicely that babies with cord ferritins preterm babies with ferritins less than 76, that should say at 36 weeks post-conceptional age, have slower central conduction speeds. So that's the myelin effect. So I think you see that the pathology that you see in the babies lays nicely onto the known neuro effects of the compound. People are very interested in long-chain polyunsaturated fatty acids, and that actually comes from a deficiency literature. It's been known for a long time that severe essential fatty acid deficiency causes hypomyelination, alters the fatty acid profile, slows the speed of processing, and these findings have been true in about every preclinical model that's been looked at. It's been much more difficult to actually show this in humans unless the deficiency is very severe. And again, this would be a whole lecture on the PUFA balance that we have in our preterm infants. but the hypothesized effects of not giving enough polyunsaturated fatty acids, we're talking about DHA, docosahexaenoic acid, and arachidonic acid, would be on myelination, again on that neuronal membrane fatty acid compositions. So the efficiency of the ability of the efficiency of synaptosomes to fuse with the outer cell membrane, and therefore synaptogenesis. Fortunately, since we've actually just in the last few weeks, a paper came out in the New England Journal of Medicine by Gould et al. That showed consistent positive effects of PUFA supplementation in preterms. And it's been difficult to show any positive effect in term babies, probably because they synthesize it relatively well from precursors, but preterms who have trouble synthesizing LC-PUFAs from the linoleic and linolenic acid backbones have benefited from PUFA supplementation. These studies need to be done long-term. They are currently underpowered to really draw any conclusions about long-term efficacy. Nevertheless, DHA really should be in the diet if it's at least 1.5% of the fatty acids. Like every other nutrient I'm about to talk about, status is not typically monitored in the unit. We really don't have good biomarkers that are easily accessible. The way we do it in the research lab is to assay the fatty acid content of the red cell membrane, because that does reflect probably what's in the brain, but that is not a clinically available test. Zinc, I really enjoy talking about zinc because I think zinc is often the forgotten nutrient. Just a kind of an aside, when you have babies that you think that are not growing, particularly not growing linearly, and you have checked everything, the baby's on adequate protein, adequate carbohydrates, adequate iron, et cetera, don't forget to check zinc. That is the hidden nutrient that is often deficient in preterm babies, and you supplement with zinc, and remarkably, those babies will often gain weight. Sodium is another one, by the way, in that bucket. But let's talk about zinc specifically in the brain. It's very important. I had showed it to you on that schematic of the cellular pathways that it regulates IGF-1 expression. So zinc deficiency decreases IGF-1 gene and protein expression. It's important in the enzymes that mediate DNA and RNA content. And then at the chemistry level, they're important for release from presynaptic boutons and for regulation of receptor function in the cerebral cortex. So it has both a direct neurologic effect as well as a regulation effect. Now, there are no studies of neurodevelopmental outcomes of preterm infants as a function of zinc status. Zinc status is actually fairly difficult to measure accurately, and it would be very difficult to isolate just the role of zinc. But we do know from studies in Peru and in Colorado that fetuses of zinc-deficient mothers, so moms are deficient, the baby is deficient, or the fetus is deficient as well, that as fetuses, they have decreased movement, they have altered autonomic nervous system stability. And then postnatally, when you follow those babies, they continue to show some neurodevelopmental issues, specifically hippocampal ones like decreased preferential looking times, although there's really no difference, for example, in the Bailey scales. There still appears to be an effect of fetal or zinc deficiency in that late fetal time period on autonomic nervous system and hippocampus. The suggested dose is 2 to 2.4 milligrams per kilogram daily. But again, like the PUFAs, we don't typically monitor zinc status. I did ask Dr. Nancy Krebs, who's a very prominent professor in this field, and her specialty is in zinc. Well, what should we do? And she said, you know, you can get a zinc level. It may be a little bit misleading, but I would treat any zinc level less than 70. Iodine. So remember I told you that iron, the three most common nutrient deficiencies around the world are iron, zinc, and iodine. If you were to eliminate them, you'd increase the world's IQ. So here's the populations at risk. There are 20 million at risk for low iodine levels worldwide, and 3 million of those will have severe CNS effects. Iodine actually has only one role as it relates to brain development, and that is in thyroid hormone. So there's no direct role of iodine itself. It simply is necessary for thyroid. And of course, we all know that low iodine status leads to hypothyroidism or cretinism, and that the earlier that happens in a child's life, let's say life being either fetal or postnatal life, the more profound the effect will be, including lower brain weight, lower brain DNA, reduced arborization, and reduced myelination. So you can see why function would be affected. Timing is absolutely critical here. So the fetal effects are much more profound than the postnatal effects. But nevertheless, even postnatal iodine deficiency due to lack of iodine in the diet will reduce verbal IQ. Again, like the others, there's no neurodevelopmental outcome studies of preterms as a function of their perinatal iodine status, and the current dosing recommendation is 30 micrograms per day. I wanted to talk just briefly about two what we call methyl donors, so folate and choline. These were not really suspected to be important in terms of neonatal brain development until very recently. And the interest in them actually stemmed from studies of folate and choline supplementation in aged adults, showing that people who were at risk for Alzheimer's or who were starting to show cognitive deficits associated with Alzheimer's improved if you supplemented them with folate. We should have been looking in the first place, as far as I'm concerned. I mean, we certainly knew about folate in the periconceptional period and the reducing the risk of spina bifida. But it's not only important in that neuroanatomy in the first 8 weeks, but also for neuronal migration. And now we know for regulation, particularly of the hippocampus in older individuals. Our concern here with the babies is that premature neonates have immature enzyme systems related to folate and require a source of folate every day. Of course, folate deficiency has been associated with neural tube defects, but more interestingly, for mental health purposes, maternal periconceptional folate deficiency is also related to the risk of autism in the offspring. So the mom's folate status as she enters pregnancy is a driver of risk for autism. There are currently no neurodevelopmental outcome studies of preterms as a function of folate status. And again, we don't tend to routinely measure that in the unit. And then maybe this is the future. Maybe we can repair certain deficits with this compound or with this nutrient, choline. Choline is of course found in many different foods. It's a complex, it's common B complex vitamin found in eggs and vegetables. We have been looking at it as a way of reversing some of the iron deficiency effects. And that's not going to be immediately obvious how that works, but it appears to work through epigenetic mechanisms. Back in the late 90s, and certainly continuing with the papers that I show you down at the bottom here, preclinical investigators have seen that choline, given at certain times during neurodevelopment, either mid-gestation or immediately in the postnatal period in rodents, improves their hippocampus, meaning their learning and memory, the brain morphology, the electrophysiology of the hippocampus. And that not only does it work in normal, sufficient animals, but it also works in rats with fetal alcohol exposure, mice with the Rett syndrome mutation, and mice with the Down syndrome mutation. So you're seeing that some sort of a very basic effect on gene expression and brain function, so in this case, learning and memory, in different species, rats versus mice, And now we have evidence in humans with fetal alcohol exposure, and in different conditions, meaning a toxin, fetal alcohol, two genetic syndromes, Rett's and Downs, and now we have shown this in rats with iron deficiency, so a nutrient deficiency. So it must be doing something fairly fundamental, and that is likely at the gene level. There are studies of prenatal choline status and neurodevelopment. They are largely mixed results. Babies or children with the top, or moms with the top quartile of second trimester choline intake, their babies, their children have better visual memory at 7 years. So that's very hopeful. On the other hand, a randomized controlled trial of choline supplementation in women didn't show much of anything in the offspring. We are running a study at the University of Minnesota, which is now out 10 years. This was in children with fetal alcohol spectrum disorder who were randomized to choline or not. And the younger children, these kids were 2 to 5 years old when enrolled. The younger children, those that still had a lot of hippocampal development potential, had improved cognition, not just 6 months and a year after choline treatment, but now 10 years out. So we're very hopeful that choline, I don't ever believe in these magic nutritional bullets, but choline seems to have the ability to change the brain's direction and function in a positive manner if given at the right time. Now, to be fair, nobody has been studying choline status or recommending dosages in preterm infants. I think that is the new world out there. So I've been talking about these long-term brain outcomes, just a couple slides to show you that there are two really major theories, and I've touched on both of them, of why or what accounts for this long-term loss of synaptic plasticity. And the first one is the critical period hypothesis. So every region of the brain, whatever it is, hippocampus, cerebellum, frontal lobe, whatever, goes through a critical period of rapid development where its nutrient requirements are very, very high. And for the hippocampus, that starts at 28 weeks gestation and goes for the first 18 months. For myelin, it starts at 32 weeks gestation. So you can see that these things happen at the time that we are involved in the baby's care. If the nutrients are not there, then we have permanent structural changes. And and the behavioral deficits that you see in adulthood, this is in rodent models, relate directly to that disordered neuronal structure. And I'm showing you here the effect of iron deficiency in the perinatal period on long-term structure. These are adult brains now that we're looking at. And on the left-hand side, we see an iron-sufficient hippocampus. These are the neuronal bodies, the bright green spots. You can see that it's very organized. And then these long dendrites hanging down, very organized, makes lots of sense, functions extremely well. These animals are very smart. The animals on the right, so these are animals that are currently, well, at the time of sacrifice were iron sufficient, but had been iron deficient during the critical period in the perinatal time frame. And you can see the disorganization of those neurons. And this is now in the adult animal. So permanent long-term changes. And these animals perform poorly, although not completely off, but poorly on memory tasks. The second possibility is, we've also talked about, which is that nutrients have the ability to change gene regulation through modification of chromatin, through epigenetics. They can be very specific genes that are important for synaptic plasticity, like brain-derived neurotrophic factor, BDNF, or for large gene networks that either code for behavioral performance or psychopathology. And there's the list. So it's not uncommon. All of these nutrients or nutritional conditions have specific effects on epigenetics of either synaptic plasticity genes or gene networks in the brain. And here's an example in an adult. This is a rat study. So these are adult hippocampuses, and the gene networks that were affected by early life neonatal iron deficiency, including risk of schizophrenia, gene networks associated with autism, and with Alzheimer's. Okay, for the last couple minutes, let's talk about the challenges. What is impeding our progress of how we know whether there's change in nutritional status in our babies, whether that's actually affecting their brain or not? Because obviously we're not doing brain biopsies or spinal taps to do nutritional status. The classic approach that people have used for years and years has been that we basically compare effects across animals models behaviorally. In other words, there are behaviors that we can measure in humans that we can also measure in animals. For example, recognition memory. Structure, well, in animals, we can certainly look directly at the structure, but in humans, we're relegated to just neuroimaging, and you can imagine how nonspecific that might be. Performance, so electrophysiology. With the animals, we can get invasive recording, but with humans, like the Siddhapa study, like Asha's study, we can record from the scalp and we can see electrical performance there, and so on. So basically, the idea here is that we tie all of these together to say that it's mostly indirect, but we have a pretty good idea of what many of the nutrients do. What we really need are what are now called bioindicators. So when you measure somebody's ferritin or you put them on a growth curve, you're looking at the child relative to population norms. But what we really want to know is how does that relationship, how does that marker tell us about the brain itself? So if you recall Sanjeev Amin study, where he showed that a ferritin less than 76 was associated with poor neurodevelopmental performance in preemies. The fifth percentile for ferritin is actually 40. It's not 76. 76 is about the 20th percentile. So relating actual numbers to actual performance is what we want to be doing. So these biomarkers that we currently measure, serum markers or growth markers, do not tell us about the effect of the nutrient on brain biology or function. And as I told you before, nutrients don't typically have signature effects on the brain. Where the research is going here is to have something that is measurable in the serum or the saliva or the urine, something that is derived from the brain or reflects what's going on in the brain, but can be measured peripherally. And this is work by Dr. Raghu Rao, who is my division head and my boss and my long-term collaborator, who's showing in a monkey model of iron deficiency that as these animals slip into iron deficiency, and if you look here down at the bottom of the table, you'll see that animals that are destined for iron deficiency, anemia, are not anemic at 2 months, nor at 4 months, only finally at 6 months. But by measuring certain biochemical events, metabolites in the serum and in the CSF at the same time, because we can do that, we can see that the brain is already changing before the anemia has set in. And that's very important, right? We want to protect the brain. So we want markers out in the serum that we can measure that are reflecting what's going on in the brain. So that's one approach. And another even more exciting approach is to get neurally derived stem cells, or neurally derived exosomes. So the body is constantly shedding little packets of what are called exosomes that contain information, and they contain proteins, they contain cytokines, they contain some messenger RNA, some metabolites. We can identify which ones are from the brain, and the hope is that from there we can start to see important compounds like brain-derived neurotrophic factor and whether it is suppressed by iron deficiency. So this technology is just starting, and I don't think this is something we're going to be using clinically, but it could tell us when these salient markers are changing, when the brain is in trouble relative to more commonly assayed serum markers like a ferritin or like a TIBC saturation. So that's where we're going. Okay, let me wrap up because I think we're going to have lots of questions here. I hope I've convinced you that through the rules of engagement here, through the interactions, that nutrient effects depend on timing, on dose and duration. timing based on when certain regions of the brain are rapidly developing and have their highest nutrient needs, and that there are certain nutrients that have a much larger impact on the early brain development. They include calories, protein, branched chain amino acids, zinc, iron, copper, etc. And then timing and dose and duration, meaning how long is the deficiency and how deep is the deficiency. Think of that as an area under the curve. So these nutrients have effects on globally on the brain or circuit specific. They can be short-term. The ones we're concerned about are the long-term ones. And most importantly for all of us, nutrition is something that we can control. We can't control hemorrhages very well. We can't control lots of things that happen to babies' brains, but nutrition is one thing that we can control. So I'm going to stop there. I realize there are many things I didn't cover, but let's have at it with the questions and maybe we can cover some of them. Dr. Manoj, back to you.
That is amazing, sir. It is, I mean, you are given an overview. Now, I'm sure there are going to be a lot of questions. I would request all the respected attendees to kindly type in your questions in the Q&A box, not in the chat box. Now, over to you, Colonel Shridhar and Colonel Subhash, who will be moderating this session. Over to you.
Thank you, Professor Michael, for a very fascinating, I think, insight into how nutrition affects the preterm brain. I think building from the basic science to how the long-term effects can actually translate into epigenetic effects or epigenetic models. It was a very, very, I think, incisive and insightful presentation, sir. There are a few questions in the chat box, sir. So the first one refers to the fact that even from iron deficient mothers, the newborns are able to extract iron. So how come iron deficiency occurs among term neonates?
Yes, great question. Thank you, Dr. Yadav. Like I said, I could have talked for an hour about iron. So there's a very interesting history with that question that goes back to the 1960s, where it was noted that babies can extract iron from their mothers. The baby is a very good parasite that way. And since the 1960s, there have been many studies done that show that the more iron deficient the mother becomes, let's say she starts with normal iron status, the more deficient she becomes, the greater the percentage of iron goes across to the fetus. So you would think the fetus would be very protected. And when the studies were done back in the 1960s, people were unable to show that the babies were iron deficient. I think it's because the mothers weren't all that deficient. The problem becomes when with a couple of conditions. So the first is maternal iron deficiency that is severe enough that the entire maternal or the maternal placental fetal triad, the entire system becomes iron deficient. And surprisingly, even though this was asked about back in the 60s, it wasn't until the last 7 years that Betsy Lozoff's group, doing a study in China, showed that there is an inflection point of maternal iron status where the fetus becomes compromised. So in other words, the fetus can draw off as much as it wants at first, but once a mom reaches a certain level of iron deficiency, the entire system is deficient and the fetus becomes deficient. And that occurs at maternal hemoglobin less than 10, and it occurs at a maternal ferritin less than 13.4. All right. So that's one way. Now, there are other ways that a neonate, a fetus, and then a neonate could be iron deficient, even with an iron sufficient mother. And that includes a placenta that does not work well. So in intrauterine growth restricted babies, about 50% of them have abnormally low iron stores at birth. Babies born to mothers that smoke, babies born to mothers with hypertension, which is one of the causes of IUGR, and babies born to mothers with diabetes, glucose intolerance in pregnancy. So all of those groups of babies also have low iron stores at birth. Now, the other point to be made here that I think is important is that these babies are not typically anemic, right? I mean, actually, the IDMs are polycythemic. They're not anemic. These are just babies born with low iron stores. But that's important because babies are going to live on those iron stores postnatally for the first 4 to 6 months, right? Because they're on a low iron diet, breast milk. And so they have to have had adequate fetal iron loading. So it's really only been in the last maybe 4 years, 3 to 4 years, that we've realized that postnatal iron deficiency, the iron deficiency we as pediatricians screen for, is set up particularly in babies who've had low fetal loading. So the other way, of course, is premature birth, and that is because babies accrete iron in that last trimester, and if you're out early, you didn't get that iron. So that's pretty simple. So there's lots of ways that babies can be iron deficient at birth. We estimate it's about 10% of the population. Sorry, that's your whole iron. That's the iron lecture.
Yeah. So I think that's the way that delayed cord clamping works, and it helps reduce the iron deficiency anemia among term babies.
Correct. Let me just expand on that. That's a very, very important point. Okay, so a baby who is born at term and whose mother did not have any of those risk gestational conditions that I just mentioned, so severe iron deficiency or moderate iron deficiency, growth restriction, infant of a diabetic mother, smoking mother, born with normal iron stores, gets delayed cord clamping. And by the way, I think we should all be calling it normal cord clamping, and the other one be called premature cord clamping. And is breastfed and is growing at the moderate rate, right? Because that's the expansion of the blood volume rate, the moderate rate of a breastfed baby compared to a formula-fed baby. That baby has enough iron to last the first 4 months. That's what they live on. And what that means is that there's going to be less and less need to give postnatal iron supplementation, which alters the microbiome, as Michael Zimmerman has shown. And so there's this kind of paradigm shift taking place that let's do the maternal care to adequately load these fetuses so they have normal ferritins, normal hemoglobins, and delayed cord clamping at birth. That should give them enough that you don't have to risk giving medicinal iron in those first few months. Important paradigm shift.
Related to iron, so if I can just pick up a couple of more questions. There is one question which asks whether iron deficiency in the first trimester affects the birth weight and the iron stores of the baby, of the fetus. And another related question is, what do you think the effect of in utero iron deficiency on the newborn? And what is your experience on latent iron deficiency at birth?
Yeah, great questions. So the answer to the first part is yes. In fact, iron deficiency in the first and second trimesters in the mother is associated with or causes low birth weight. And low birth weight is defined in this case as either intrauterine growth restriction or preterm birth. But either way, you get a baby less than 2,500 grams. So there's a trial going on right now of giving iron, in this case IV iron, to mothers who are moderately iron deficient. This is taking place in Belgavi and in Jaipur. So these are moms with hemoglobins of say 7.5, 8, something like that at the end of the first trimester, to see if we can reduce the rate of low birth weight and also improve the iron status and neurodevelopmental outcomes of the kids. So spot on for that one. Do babies who are iron deficient at birth have neurodevelopmental sequelae? Yes. They have poor recognition memory. That's Asha Sidipa's study. Betsy Lozoff has since replicated that in a group in China. And those children, when they were followed up, continued to have recognition memory, so learning and memory issues, until about the age of 5. And then it got better. and which is wonderful, except that they then had frontal lobe problems, meaning attention problems and planning problems when they were 10. So that's, again, I think that idea of this patterning that takes place either through critical periods or epigenetics, we don't know, but you can see, you can observe that effect in the humans. So yes, those babies, to be honest, I think that part of why an infant of a diabetic mother is so difficult to take care of frequently, especially with their heart failure or heart issues, is their tissues are iron deficient. They're not performing well.
Just a couple of more questions on iron, then I'll hand over to my co-chair. There is one question which says iron increases the incidence of some infections, as we all know, given early for maybe 2 weeks. And the second one, sir, is do we need to monitor serum ferritin levels in a preterm or an at-risk neonate? If yes, is there an appropriate time for monitoring? Your comments, sir.
Yes. So yes, for the first part, iron and infection. Iron is known as the bad boy of nutrition. But I think all that says is like literally every nutrient, it has a U-shaped risk curve, right? And so adequacy gives you the lowest risk. Deficiency is what we've been talking about. That gives you certain risks to the brain, to function, and so on. But overload or excess also causes issues. And approximately the most important one is infection. And the evidence for that in babies has been that iron supplementation, this is of term kids, term breastfed kids, iron supplementation shifts the microbiome toward a more pathogenic one, more E. coli and more salmonella. And I think that the person who's done this work, Michael Zimmerman, I think he's also shown more diarrheal disease, but don't quote me on that. I'm not 100% sure on that. The more obvious one is in the slightly older child, and that's with malaria. I mean, malaria loves iron. So malaria is sitting inside the red cells, and the iron is going in there to make hemoglobin, and the malaria is saying, thank you very much, I'll multiply. So we need to figure out ways, and fetal loading is one way to protect that young child from the potential infection issues of giving supplemental iron. Remind me the second question.
So the second question was, do we need to monitor serum ferritin levels in either preterm or at-risk units?
Yes, absolutely. In fact, so I don't get to make the policy decisions. I wish I did. But, each state in the United States has a panel of cord blood screening that they do. I don't know if there's universal screening in India or not. Of all sorts of genetic disorders, and that's wonderful. We should know who's got PKU and who's got thyroid problems, But my goodness, iron deficiency affects 2 billion people, mostly women and children. Why are we not monitoring that? Just to know where we're starting from and know when we should start to supplement those kids, given what we just talked about. So that's just term kids. In preterm kids, yes. So here's why. First of all, preterms, because they did not get their third trimester loading of iron, right, because they came out early, have lower iron stores to start with. Secondly, we then bleed them, which is a major drain on iron. Every gram of hemoglobin you take off the baby is 3.5 milligrams of iron. And of course, we do transfuse them, but remember, we transfuse them once we've bled them down. It's not like we transfuse them first and then bleed them. So you're always behind the 8 ball. When we have measured ferritins in preterm babies, there's a much wider range of status from zero, like zero ferritin, no stores at all, to thousands, 3,000, 2,000 post-transfusion. And so this wide range, I think, needs monitoring so that you know what to do in terms of dosing. And I might comment that we use, and again, Dr. Raghu has set the standard here of when we monitor. So we start at 2 weeks of age, and we monitor ferritins every 2 weeks in the NICU, and we respond when the ferritin is declining. If your ferritin is going down, it is because you don't have enough dietary iron. You're now having to use your storage iron, right? It's just like money in the bank. So when the ferritin goes below 100 micrograms per liter, we increase the dose by 1 per kilo per day. If we stumble across a baby who is below 40, which is where the neurologic risk is, we go up by 2 milligrams per kilo per day. So start monitoring at 2 weeks of age, monitor every 2 weeks.
Thank you, sir. Now I hand over to Colonel Subhash, my co-chair, for the rest of the questions.
Good morning, Professor Michael.
Hi.
A few more questions for you. One question is that is it safe to give aggressive parenteral or enteral nutrition in sick preterm babies? Is there any possibility of nutri-trauma with aggressive feeding?
Yeah, that's a very good question, and one that's been debated for a long time. I think certainly as I look at 35 years of career in nutrition, we've learned to be more aggressive over time. I think the concerns about necrotizing enterocolitis, does feeding cause necrotizing enterocolitis, is metabolic acidosis from large amounts of amino acids and so on. I think we've got the parameters pretty well set, and work by Professor Hay at Colorado, along with Professor Thurine, showed that it is safe to start protein, for example, at 3 grams per kilo per day and move up to around 4. But as I said, every nutrient has a U-shaped curve, and I think there are a couple studies out there that bear looking at where people have thought, well, if some is good, more must be better. Let's give 4.5 grams per kilo or 5 grams per kilo. And yes, you do cause nutri-trauma there, and you get smaller head circumferences, and there are issues. So I think there are boundaries to it, but I think we're better defined in terms of what the targets should be. On the whole, if you have a nutrition support service, or if you're analyzing nutritional intake in our premiums, we typically undershoot what the targets are. For a while, it's a great physiologic question by Dr. Kumar, because in the adult literature, in the adult critical care literature, if you give a lot of carbohydrate, you're trying to give more calories because the person is sick, you increase the oxygen consumption of the cells dramatically, and you increase the RQ, so you're generating more CO2. So if you're trying to get somebody off a ventilator and you're giving them pure carbohydrate calories, you're actually generating more CO2 because the RQ is 1.0, as opposed to using more fat or more protein. So I think there's some nuancing that takes place in terms of not just how much you're giving, but the mixture of what you're giving as well. So it's a great question, and it's an area that just hasn't been studied enough because we've been so concerned on the deficiency side. But as we're now kind of reaching what we think are optimal regimens, we need to make sure that we're not thinking, well, if some is good, more is better. More is probably going to cause nutri-trauma. Good question.
Well, another question is, how long the nutritional supplements to be continued? Like, when should I stop supplementing nutrients for a growing preterm? That's the question.
Yeah, that's an outstanding question too. And it reflects the fact that until, at least in the States, very recently, those who did the neonatal care did not do the neonatal follow-up. And so it was like, once you're discharged, well, that's somebody else's problem to deal with. And that's a very short-sighted thing, because when you think about it, babies are leaving the intensive care unit at around term, right? They leave around, say, 36, 37 weeks, and yet they're clearly not the size of a fully grown in utero term baby. And what that means is, so let's imagine that a baby leaves, a term baby, let's say, is 3 kilos, 3.3 kilos. It needs to triple its birth weight by a year, right, to get to 10 kilos. A baby who leaves the NICU, having been undernourished and all the stuff that we see, right, leaves the NICU, let's say, at 37 weeks at 2.2 kilos or 2.5 kilos. That baby needs to quadruple, almost quintuple its so-called birth weight or discharge weight in that first year. And so the nutritional demands are much, much higher. You can then assess which nutrients are the ones that are at risk. So the first and most obvious is calorie and protein. You see that in that essentially EUGR, extrauterine growth restricted baby that you're sending home. But the other nutrients at risk are iron, calcium, so your bone density, and those are the main ones, I think. There are recommendations, they're very hidden in the literature, of following up nutritional status, not just with growth curves in the outpatient setting, but also iron status and alkaline phosphatase, or some way of looking at the calcium phosphorus status, at about 6 to 8 weeks out, and then again at 6 months out. Very, very, very few pediatricians in my country or NICU follow-up clinics, including mine, actually do that. So I think it's a really, really good question. The answer is, for sure, they should be going home on supplements. And we send many babies home, even if they're, and most of them are being fed mom's milk, but we fortify that with formula powder to give those specific nutrients. So calcium, iron, protein, and energy, at least for the first. I usually see them back at 4 months corrected age. So it's usually until at least 4 months corrected age.
Thank you. Now, again, some questions on iron, using ferritin level for anemia of prematurity and iron supply to preterm. Is that your practice, and what cutoff you use for ferritin, both for a low ferritin and high ferritin? That is, like, if the ferritin is high, when do you stop supplementing iron? That's the question.
Yeah, excellent question. So the answer is yes, we do follow those. My friend, Professor Jack Widnes, who was at Iowa and studied anemia of prematurity for as long as I studied iron, maybe longer, said we should stop calling it the anemia of prematurity and call it the anemia of phlebotomy, which is probably accurate, right? So I think the principle to remember, the nutrient principle to remember here, is that iron, when you supplement it, will go to the red blood cells. It goes to the red blood cells before it goes to the brain. It goes to the red cells before it goes to the heart. It goes before it goes to the skeletal muscle, and it certainly goes to the red cells before it goes to the liver, which is, the ferritin stores. So when we are monitoring babies for their iron status and anemia in the NICU, we are measuring, starting at 2 weeks, we're measuring a hemoglobin, a retic count, and a ferritin. So for the cutoffs, if the ferritin, and usually we will have started iron therapy at 2 weeks of age. We're waiting for 2 weeks in that case because we want antioxidant systems like vitamin C to mature. So we start at 2 weeks of age, and we start them on a dose somewhere between 2 and 4 milligrams per kilo per day. So then we measure 2 weeks later, and if we see that the ferritin is dropping, right, then we will increase the iron by 1 per kilo, particularly if it's less than 100. On the other hand, the question about, well, what if they've been multiply transfused, they've got a lot of ferritin on board, do we stop the iron? And yes, I think the answer is just simply yes, we do. And we wait until the ferritin comes down to 350 or less, and then we restart. I will just say that there's so little iron that we give enterally compared to what you get in a red cell transfusion that you could choose not to stop the iron just so you don't have to remember to restart it. It's probably not going to get absorbed anyway. But technically, in a paper that Magnus Domelov and I wrote back in 2015, we recommend stopping iron if your ferritin is above 350, and then restarting it once it drops back down.
On the same line, Dr. Ashok Kumar also asked that probably he listened to your lecture in Bangalore, and their unit started monitoring serum ferritin in preterm babies starting at 2 weeks. So they found many cases had ferritin above 500, and some values were even in thousands. So without monitoring serum ferritin, probably you would not have realized this. Your comments on this? Probably you have already commented that above 300, we should probably not be giving iron.
Correct. Yes. So those, we're still investigating. There are multiple studies out there that are ongoing investigating. So why do those babies have ferritins of 500 or 1,000? I've seen them as high as 3,000. And I think there are 2 reasons. One is multiply transfused. So you're not always using red cells red cell transfusions just because the baby's anemic. We use red cell transfusions for blood pressure support in critically ill babies and so on. So you can iron load a baby pretty quickly, right? In those cases, if there is no let me just say, it could be iron loading, but it can also be ferritin acting as an acute phase reactant. And we know that transfusions, more or less, depending on the sex of the baby, depending on its immune constitution, you can get some pretty big inflammatory responses after a red cell transfusion. So when you get a ferritin of 500 or 1,000, it might really reflect how much iron is in there, or it might just be ferritin acting as an acute phase reactant. In either case, the proper thing to do is to stop the iron, the enteral iron, and wait until it's down to 300. Either the inflammation is resolved or they've mobilized that iron. And I will say that since we started monitoring ferritins every 2 weeks, and that's been a few years now, I've been surprised at how quickly that number can come down. You know, you sit there and you look at a ferritin of 1,000, you think, oh, well, I've got weeks to wait. Not really. It could be down in a week or two. So it just is a way of reminding to continue to monitor.
Well, one question is on human milk fortifier, that is HMF, human milk fortifier. So most of the units in India would be starting HMF when the baby reaches 100 ml per kg of express breast milk. So what is your unit protocol or your suggestion to add HMF? Like, do you also start at 100 or some other?
Yeah, somewhere around there. We've surveyed the units nationally on that question, and the answer is anywhere from 40 per kilo to full feeds at 150 per kilo. And we're pretty much in the middle, right around 100 per kilo. You know, you got to figure a couple things about the dynamics of iron if that's what the question is about, iron, and not so much about fortification itself. Well, let me just say, fortification itself is probably safe very early as the baby is working its way up. So there's it's always been a lot of debate about hyperosmolality and so on. I don't think anybody thinks that that's a real issue. So you could start fortifying theoretically at 40 per kilo or 60 per kilo. I don't know if there's a Cochrane analysis of that or not. Nevertheless, for the iron portion of it, because the fortifier, one of the fortifiers at least has iron in it, you typically don't need a source of enteral iron for your first 2 weeks anyway. So by that time, you've probably already built the baby up to pretty close to full feeds, and the baby is starting to grow. Again, the biggest pressure on the iron system is blood volume expansion, which is equal to, proportional to growth. So people get very worked up about iron, not getting enough iron in the first couple of weeks. I don't think that's a big issue. I think it's after 2 weeks when you've got a growing preemie that you really want that supplementation.
One very interesting question is that should the preterm reach iron deficiency anemia state to have effect on brain development, or just simply low iron level is good enough to negatively impact the brain development?
Yep, good question. So one of the principles that my group happened to discover was that iron is prioritized to the red blood cells over the brain. So anytime you're anemic and you give iron, it goes to the red cells, not over the brain. And it goes to the brain over the heart, and it goes to the heart over the skeletal muscle, and so on, the hierarchy. Professor Sunny Jewell, who is at University of Washington, has stressed, and the peanut study shows you, that you got to give a lot of iron to fill both of the buckets. So both the red cell bucket and the brain bucket. And that number in her study, especially if you're using EPO, which of course is driving red cell iron, or driving iron into red cells, that number might approach 12 milligrams per kilo per day, which I think just makes all of us hold our breath. That just seems like so much iron. And she's actually starting a trial of IV iron. Maybe that's a better way to go, rather than oral iron. So it takes a lot to fill both of those buckets, and just realize that the more you phlebotomize the baby, and of course we have to do this, but the more you phlebotomize the baby, the bigger the hole, the bigger the sink is, that when you put iron back in, it goes into that sink and doesn't go to the brain. So I'll give you an example. Infants of diabetic mothers, all right? These are babies who are polycythemic in utero. They're nowhere near anemic. But they have taken, because they have a high erythropoietin state in utero, and that's driven by hyperglycemia, hyperinsulinemia. Because they have a high EPO level chronically in utero, they have a big red cell mass. Their brains are iron deficient. We've done autopsy studies, and we've looked at ferritin concentrations, and they are rock bottom because of that drive to shunt iron into the red cells at the expense of the brain. So yeah, you got to give a fair amount of iron to fill both buckets when you've got your preemies, especially those that are having a lot of phlebotomy.
May I request Dr. Shridhar, my co-moderator, to take the rest of the questions?
Sir, there is one question again related to the dosing of iron. When should we add iron in a term baby with latent iron deficiency? Someone who has about 11 to 75 nanograms per ml at birth.
That's a good question. So a term baby who latent iron deficiency, I assume, means has a normal hemoglobin but a low ferritin. Yeah. I will be perfectly honest, there are no recommendations, currently official recommendations, to treat low ferritins. There aren't. If an older child happened to be found that way, I know what the advice in the clinic would be. It would be give that child more iron-rich foods. So it would be a kind of a food-based approach. So I can't officially say that a baby who's got latent iron deficiency, though I know that's affecting the kid's brain, I can't officially sit here and say, well, that's an indication to treat that baby. But I think I probably would treat that baby. And I would start at 2 weeks of age. Again, I want the antioxidant systems to mature. There was a conference at the National Institutes of Health back in 2016 that is published in the 2017 American Journal of Clinical Nutrition that's worth looking at, where they specifically convened a panel of whether a non-anemic or not, whether iron's sufficient, but what they really meant was non-anemic mothers, pregnant women, or children should be supplemented with iron, or if that's a risk. And you could read the 17 articles that are there. It is all over the map. So there's just nothing official. My inclination is, if you're treating a population, if your population in your clinic tends to be an iron deficient population, I would be treating a low ferritin before the kid becomes anemic, because I know the brain would be at risk.
Important message, sir. So I have one last question before I hand over to Professor PC Manoj. We all know the Barker hypothesis, and many of us also know that the standard recommendations of the ESPGHAN, the American Academy of Pediatrics, are to meet the RDAs. But there is always this uncertain, maybe an uneasy feeling, how much is really the limit? Though there are guidelines that are given, the catch-up growth is good, but is it something that we're doing without realizing what the long-term effects are? Is body composition the way to go? So your comments on this, sir?
Oh, that's also right at the cutting edge of research question. Let me break that down into a couple of places. So the Barker hypothesis initially was a demonstration that low birth weight was associated with a higher risk of hypertension and diabetes in people that live in England 60 to 80 years later. The refinement of the hypothesis was, it's not so much how small you were at birth, but if you grew rapidly after having been small. And that's the whole point, as we were talking about earlier, about the fetus, right? Trying to anticipate what life postnatally is going to be like, because I'm setting metabolic enzymes. And it would make sense, if you will, that a growth-restricted fetus probably is going to be also nutrient limited postnatally. So let's set everything thrifty. But instead you come out and boom, you get fed lots and lots of food. And then you get central obesity and hypertension and so on. So one question has been, with our preemies who go through a similar period of pretty severe growth restriction in the NICU, right? Because they're sick and we can't feed them and so on. And then they get better. And what do? Get them catch-up growth, right? Feed them, get them up there. Are we increasing the risk of cardiovascular disease in those babies? And there was some data from Scott Denny's group in Indiana that showed insulin resistance as early as 18 months in extremely low birth weight preemies, the kids less than 1,000 grams. That's still out there. That's not been proven. And I think we still are concerned. My colleague, Professor Sarah Rammel, does work on that. And she is, first of all, she's the one, apropos of your body composition question, she's the one who's shown that we should be looking at growth as linear growth. That's the solid protein growth, if you will, that is the same as the structure of the brain, the structure of the lungs, organ maturation. So not so much about weight gain. And she's the one who's shown that it's linear growth that's associated with neurodevelopmental outcome more so than weight gain, right? So that's a whole different problem to solve in terms of how do we get better linear growth. But body composition would be one way to be assessing that question. What Sarah tells me, and I trust her because I think her research is good, is that catch-up growth is really important prior to term, because that's going to protect the brain. But too rapid a growth in that first year of life alters the body composition at least until the age of 4 and may constitute a cardiovascular risk. And I think that's as close as we can get right now. I know she has a grant with Mandy Balfour at Harvard, just getting started on, apropos of Dr. Kumar's question about aggressive nutrition, about using aggressive nutrition to foster that early catch-up growth. And they are measuring both neurodevelopment as well as cardiovascular outcomes. So maybe we'll get some answers there. It's a great question. I think it's a matter of timing. I think you want your catch-up growth to be early, but not be pushing on the accelerator quite so hard once they're gone, once they're discharged. That's my best answer, I think.
Thank you again for that fascinating insight into nutrition affecting the preterm brain. Now over to Dr. Manoj, sir.
Well, thank you so much, Professor Michael Georgieff. As usual, it was an amazing lecture, and as we suspected, we are finding it difficult to cut short the questions and wind up, but then we have exceeded our time, so we'll stop today. It was so nice of you, Professor Georgieff, to find time to spend with us. I sincerely wish that in the next physical conference we host, you will be able to spare time and join us here in India.
That would be wonderful. And I feel so honored to be considered a legend. I have to say,
you are,
you're a legend in your own mind.
All counts you are. Just now, when like Professor Bangalari, who was here, and a few others were all remarking the same thing. It's your humility that speaks. So thank you so much, sir. I would also like to thank the moderators for today, both from the armed forces, Colonel Shridhar and Colonel Subhash, who had handled the very difficult task of compiling the questions and then getting the gist of hopefully whatever the delegates wanted to know. Thank you so much, both of you. And I would also like